Hi students, in this video we are going to learn the second part of the lesson, Health and Diseases. Let us learn the basic concepts of immunology. What is immunology? The study of immune system is immunology. Immune system protects the individual from various infection. Immunity refers to all the mechanism used by the body to protect against disease-causing pathogens. When the immune system does not function efficiently, it leads to the disease in the individual. The overall ability to fight against the disease-causing pathogen is called immunity. This is the definition of immunity. What is immunity? The overall ability of the body to fight against any disease-causing pathogen is called immunity. This immunity is otherwise called as disease resistance, and the lack of immunity is known as susceptibility. Immunity is highly specific. Normally, our immune system will work for the destruction and elimination of the invading organism. And these immune reactions which are produced by our immune system are destructive in nature, and they are made only in response to the foreign molecules and not to the molecules of the host itself. And so, the ability to distinguish the foreign molecules from the self is another fundamental feature of the immune system. The ability to distinguish between the foreign and the self molecules. Sometimes what happens, our body immune system fails to distinguish between the foreign and the self molecules, and it reacts destructively against its own molecule, against the host own molecule, and such kind of diseases are called autoimmune diseases, and that could be fatal to the organisms. Almost this antigen, the foreign bodies are macromolecules like proteins, polysaccharide, nucleic acid, etc. So as long as they are foreign to the organism body, recipient organism, they can induce an immune response in the body. Any substance that is capable of eliciting immune response is called as antigen. If it is able to produce an immune response in the body, then that substance is called as antigen. Antigen means antibody generator. There are two broad classes of immunity. They are innate immunity and adaptive immunity. Adaptive immunity is otherwise called acquired immunity. What is this innate immunity? So innate immunity is present in every individual's body right from the birth. Every individual possesses this innate immunity right from the birth. It is a natural phenomenon to fight against the infection. And so the innate defense mechanism are non-specific. They are not specific to a particular antigen. They work against a wide range of potentially infectious agents. And so it is otherwise known as innate immunity is otherwise known as non-specific immunity or natural immunity. The number of defense mechanisms will be operated non-specifically against a large number of microorganisms. Now what are the types and mechanisms of innate immunity? There are four types of innate immunity. They are anatomical barrier, physiological barrier, phagocytic barrier, and inflammatory barriers. Anatomical barriers, the term itself refers that it is by the structure itself, the structure of the body itself gives some barriers to this foreign pathogens, foreign agents. They are skin and the mucous membrane. The skin will prevent the entry of microbes from outside. The skin is maintained at the acidic environment of about pH 3 to 5, and this acidic environment will retard the growth of the microbes, will not allow the microbes to grow. And the mucous membrane, the mucus which is produced by this mucous membrane will entrap the foreign microorganism. It will not allow the microorganism to proceed further to attach themselves in any other tissue of the body. Instead, these mucus will entrap the foreign microorganism by their sticky nature. What are the physiological barriers? Temperature, low pH and chemical mediators. Temperature, the normal body temperature will prevent the growth of the pathogen. Normal body temperature of the organism will not allow the growth of the microorganism. And fever also inhibit the growth of pathogens. Whenever there is an infection, temperature rises and cause fever as a preventive mechanism to prevent the growth of the pathogen. Secondly, low pH, acidic nature of the gastric secretion like HCl will kill most of the ingested microbes in the stomach itself. And the chemical mediators like lysozyme. So lysozyme act as an antibacterial agent. And these lysozymes will cleave the bacterial cell wall, will destroy the bacterial cell wall, and the interferons induce the antiviral state of the uninfected cells. Interferons will act against the virus, and they are the antiviral substances which will not allow the virus to infect many more cells. Thirdly, complementary substances which are produced by the leukocytes, and these substances will lyse the pathogenic microbes and they will facilitate phagocytosis. Phagocytosis means cell eating. They will engulf the pathogenic microbes and they will destroy the pathogens by phagocytosis. Thirdly, phagocytic barriers. Specialized cells are present in our immune systems like monocytes, neutrophils and tissue macrophages. And these cells will phagocytose, that means they will digest the whole microorganism by engulfing them, which is called cell eating. Fourthly, inflammatory barriers. When there is a tissue damage or when there is an infection, it will induce the leakage of the vascular fluid from the vascular tissue, and this tissue will contain chemotactic signals. They will produce the chemotactic signals like serotonin, histamine and prostaglandins. Whenever there is a damage in the tissue will induce the leakage of the vascular fluid and produce a chemotactic signals like serotonin, histamine and prostaglandin. This will make the phagocytic cells to flow into the affected area, and this phenomenon is known as diapedesis. When there is an influx of this phagocytic cell into the affected area, and it causes inflammation in the region of tissue damage or infection. The second type of immunity is called as acquired immunity, and this immunity is acquired by the individual after birth, and so it is known as acquired immunity. It is the body's resistance to a specific pathogen, and so it is called a specific immunity. The unique feature of the acquired immunity is antigenic specificity. It is specific to a specific antigen. And diversity. Secondly, diversity. They are diversified. Different types of immunity molecules will act upon different types of antigens. And recognition of the self and the non-self. And immunological memory, and it causes a memory in the body of the organism. Components of the acquired immunity. Acquired immunity has two components. They are cell-mediated immunity and antibody-mediated immunity. This antibody-mediated immunity is otherwise called as humoral immunity. What is this cell-mediated immunity? So when the pathogens are destroyed by the cells, without producing antibodies, the cell itself, when it is capable of destroying the pathogen without antibody production, then it is known as cell-mediated immune response or cell-mediated immunity. And this is brought about by the T cells, macrophage cells, natural killer cells, in our body. Secondly, antibody mediated immunity or humoral immunity. In this type of immunity, there will be production of antibodies against the antigen, and so it is known as antibody mediated or humoral immunity. And this is brought about by the antibodies are produced by the B cells with the help of the antigen presenting cells and the T helper cells. So antibodies are produced by the B cells, and with the help of the antigen presenting, these antigen presenting cells will capture the antigen with the help of the T helper cells, and they will present this antigen to the antibodies, and antibodies will destroy the antigen, attaching themselves to the antigens. Antibody production is the characteristic feature of the vertebrates only. They are not produced by invertebrates. What are the types of acquired immunity? The acquired immunity may be active or passive immunity. What is this active acquired immunity? This is the immunological resistance developed by the organism through the production of the antibodies in their body. This is called active immunity. So active immunity is acquired through the use of person's immune response, and this will produce the memory cells. Active immunity is produced because of an infection or by immunization also. Passive immunity does not require the body to produce antibody to the antigen. The antibodies are introduced from outside into the organism. Thus the passive immunity is acquired without the activation of the person's immune system, and therefore there is no memory. This is the schematic representation of the acquired immunity. It can be classified into active acquired immunity and passive acquired immunity. Active acquired immunity is produced by the immune system of the individual's body, whereas passive immunity has no role to play by the individual's immune system. And active immunity is classified into natural and artificial. So naturally, the immune response will be generated by the individual's body against the pathogen when the pathogen is invading into their body, and it can be artificially created by administering vaccination or immunization. Whereas in the passive immunity, it is naturally done by the mother's milk colostrum, IgA immunoglobulin, and the fetus receives the immunoglobulin through the placenta from the mother. Artificial passive immunity is by administering the anti-tetanus serum or anti-diphtherial serum. Administering the plasma therapy, the serum which contains the antibody is injected into the individual's body to develop the passive immunity. What are the differences between active and passive immunity? Active immunity is produced actively by the host immune system by the infection or the introduction of the antigen, whereas the passive immunity is received passively and there is no role of the individual's immune system. The host participation is not there. Active immunity is produced due to the contact with the pathogen or by the antigen, whereas the passive immunity is produced due to the antibodies which are obtained from outside. They are injected into the individual's body, and so the active immunity is durable. It lasts long and it is effective in protection. It protects the individual to a greater extent, whereas the passive immunity is transient, means temporary. It depends on the individual who receives it, and it is less effective. And the immunological memory will be present in the active immunity. So once the individual got the disease, again the individual will not be infected by the same pathogens. Even if they come in contact with even if they encounter the same antigen, they will give a heightened immunological response and fight against the disease. They will not be getting the disease again. Whereas in the passive immunity, there is no memory cells, no memory is created in the passive immunity. A booster effect on the subsequent dose is possible. So we can give the booster vaccination, by which we can increase the immunity in the individual's body, whereas subsequent dose will not give much effect in the passive immunity. In active immunity, immunity is effective only after a short period. whereas in the passive immunity, immunity develops immediately. So in the active immunity, the antigens are introduced into the individual's body, and it makes the host immune system to produce the antibodies for the antigen, whereas in the passive immunity, the antibodies which are prepared outside or which is taken from the individual who has recovered from the disease will be administered into the patient's body. Now, what is hematopoiesis? Production of the blood cells by the bone marrow is called as hematopoiesis. Now let us see the immune responses. The immune responses may be primary or secondary. Primary response occurs first. Primary immune response occurs when the pathogen comes in contact with the immune system of the body for the first time. And during this time, the immune system will learn to recognize the antigen. So what type of antigen has entered into our body. Then our body will produce the antibody against it. And eventually it will produce the memory lymphocytes also for it. So here we can see the pathogen which is entering into the body, and it will be encountered by the B cells first. These B cells will engulf this pathogen, it will break down the pathogen, and it will present the pathogen to the T cells. And these T cells will bind to the pathogens, and it will send a chemical signal alerting all the rest of the immune system. And the B cells will divide into two types of cells, namely the memory cells and the plasma cells. And the plasma cells will release the antibodies against this antigen. And these antibodies will destroy the pathogen. The memory cells will remain inactive until the same pathogen is encountered again. So once again, when the pathogen encounters the same person, this memory cells will be triggered and give a secondary immune response. So memory cells will produce the plasma cells at a faster rate than the primary immune response and the secondary immune response. Memory cells will produce the plasma cells in the secondary immune response at a faster rate. Now the secondary immune response. Secondary immune response occurs when the person is exposed to the same antigen for the second time and again and again. So during this time, the immunological memory has been established. So already in the primary immune response, the immunological memory has been established. And so the immune response can start producing the antibodies immediately. So it will not take the time for the recognition of the antigen and the production of the antibodies. It will be very immediate, and quickly it will start producing the antibodies. So within an hour after recognition of the antigen, a new army of the plasma cells will be generated, and within 2 to 3 days the antibody concentration in the blood will rise steeply, and it reaches much higher level than the primary response, and so it is called as booster response. Differences between the primary and the secondary immune responses. The primary immune response occurs as a result of the primary contact or the first contact with the antigen. The secondary immune response occurs as a result of second and the subsequent contact with the same antigen. In the primary immune response, the antibody level will reach the peak within 7 to 10 days, whereas in the secondary immune response, the antibody level reaches the peak within 3 to 5 days itself. It takes a longer time to establish the immunity in the primary immune response, whereas the immunity establishment takes place within a short time in the secondary immune response. There will be a rapid decline in the antibody level after primary immune response, whereas the antibody level will remain high for long time in the secondary immune response. Primary immune response mainly happens in the lymph nodes and the spleen, and the secondary immune response appear mainly in the bone marrow, and that will be followed by the spleen and the lymph nodes. Next we have lymphoid organs. The immune system consists of several lymphoid organs and tissues. They are differing in their structure and their function. and they are distributed widely. They are widely dispersed in the body. The organs which are involved in the origin, maturation and proliferation or multiplication of these lymphocytes are called as lymphoid organs. So which are called lymphoid organs? The organs which are involved in the origin, maturation and proliferation of the lymphocytes are called as lymphoid organs. Based on their function, they are classified into primary or central lymphoid organ and secondary or peripheral lymphoid organ. The primary lymphoid organ will provide suitable environment for the lymphocytic maturation, for the maturation of the lymphocytes, whereas the secondary lymphoid organ will help in trapping of the antigen and makes them available for the mature lymphocytes to attack and kill these antigens. The primary lymphoid organs are bursa of Fabricius in birds, bone marrow and thymus glands in mammals. So they constitute the primary lymphoid organs, and they are involved in the production as well as early selection of the lymphocytes. The lymphocytes which are produced out of these primary lymphoid organs are dedicated to a particular antigenic specificity. Only when the lymphocyte get mature in the primary lymphoid organs, they become immunocompetent cells. Then only they can do their function appropriately in our body. In mammals, the B lymphocytes or B cells maturation occur in the bone marrow. So you can remember as B for B. B cells mature in the bone marrow, as well as T cells mature in the thymus, T for T. T lymphocytes mature in the thymus gland. Now let us see the first gland, that is thymus gland. So thymus gland is a flat bilobed organ, and it is located above the heart behind the sternum in our chest. Sternum bone is the breastbone. It has two lobes, and each lobe of the thymus contains many lobules. Numerous lobules are present in the thymus gland, and they are separated from one another by the connective tissue which is called as septum. In plural it is septa. And each lobule is differentiated into two compartments. The outer compartment is called as cortex, and it is densely packed with the immature T cells. Immature T cells called as thymocytes. Whereas the inner compartment, which is called medulla, is sparsely populated with the thymocyte. The main function, the main secretion of the thymus gland is the thymosin hormone. And this thymosin hormone only stimulates the T cells to become mature and immunocompetent T cells. So at the early teenage, that is around 12 years of age, the thymus gland will begin to degenerate, will begin to atrophic condition. That is, they will degenerate, and with this thymus gland will be replaced by the adipose tissue, and thymus gland will be absent in the adult individuals. They are present only in the young children. It is present and most active during the neonatal and the pre-adolescent periods. The next lymphoid organ is the bone marrow. Bone marrow is a lymphoid tissue found within the spongy portion of the bone, that is inside the bone. The bone marrow contains stem cells, and they are known as hematopoietic cells. And these cells have the potential to multiply through the cell division. Because they are stem cells, they are always in the state of proliferation. They divide and divide, produce many number of cells, and they can differentiate and mature into different kinds of blood cells, like red blood cells, platelet cells and white blood cells. The secondary or the peripheral lymphoid organs are used for trapping the antigen. They will help to localize the antigen so that these antigens can be effectively exposed to the mature lymphocyte to do their function. The best examples of the secondary lymphoid organs are lymph nodes. They are present all over the body. Appendix at the junction of the small intestine and the large intestine. Peyer's patch of the gastrointestinal tract. They are present in the gastrointestinal tract all along. Tonsils at your throat region. adenoid, see the adenoid, the location of the adenoid. Spleen is present in the abdominal cavity beside the stomach. Malt is mucosal associated lymphoid tissue. Gut associated in lymphoid tissue, that is gut, the intestine, our alimentary canal. Balt, bronchial or tracheal associated lymphoid tissue, that is in our respiratory tract. What is this Peyer's patch? Peyer's patch are the oval shaped structure, and they are thickened tissue. They are embedded in the mucus secreting lining of the small intestine of our body. They are present in the small intestine, and the Peyer's patch will contain a variety of immune cells, including macrophage, dendritic cells, T cells and B cells. Similarly, the tonsils are present in the palatine in our mouth, in the upper jaw. On the roof of the upper jaw, a pair of soft tissue mass located at the back of our throat, and the tonsils is a part of lymphatic system. They help to fight against the infection, including bacteria and viruses that enter through our oral cavity. Spleen is the second lymphoid organ. It is located in the abdominal cavity close to our diaphragm and the stomach. The spleen contains B cells and the T cells, and this also brings about the humoral and the cell mediated immunity. And the adenoid or the glands which are located in the roof of our mouth and behind the soft palate, in the junction of the nose and the throat region. The adenoid produce antibodies that will help to fight against the infection. So adenoid will shrink during the adolescent and may disappear in the adulthood. So adenoid, as the thymus gland, adenoid gland also will disappear during the adolescent stage, and they are not present in the adulthood. The next secondary lymphoid organ is the lymph node. Lymph node is a small bean-shaped structure and becomes a very important part of the body's immune system. It is the organ that encounters the antigen that enters the tissue spaces first. The lymph node will filter this antigen because it is tightly packed with the white blood cells, namely the lymphocytes and the macrophages. So when the antigen pass through the lymph nodes, they get trapped inside, and the antibodies are produced by the B lymphocytes. There are hundreds of lymph nodes found in our body, and they are distributed all over our body, and they are connected to each other by lymph vessels. So in this picture you can see the lymph nodes and the lymph vessels. What is lymph? Lymph is a clear, transparent, colorless, mobile, extracellular fluid connective tissue. It is clear and transparent and so no color, and they will move about, so mobile, and they are out of the cell, they flow out of the cells, so extracellular fluid connective tissue. When the lymph percolates, when they pass through the lymph nodes, these lymph will bring the antigen to the lymph nodes, which can be trapped by the lymph nodes by the phagocytic cells, follicular cells and interdigitating dendritic cells. So this is the dendritic cell which has the finger-like projections outside, and so it can trap the antigen very easily. They engulf the antigen, either bacteria or virus, and they will destroy it through phagocytosis. Lymph node has three zones. They are the outer cortex, inner to that paracortex, and the middle region medulla. The outer cortex zone consists of the T lymphocytes and interdigitating dendritic cells. Similarly, the paracortex, which is inner to the cortex, contains T lymphocytes and the dendritic cells. The innermost medullary region is also sparsely populated with the lymphocyte, but they contain more of the plasma cells which will secrete the antibody molecules for the trapped antigen. When the lymph enters through the afferent lymphatic vessels, so these are the afferent lymphatic vessels, the antigen is brought inside the lymph nodes. It will pass through the cortex, paracortex and medulla, and it gives sufficient chance for the phagocytic cell and the dendritic cells which are present in the cortex, paracortex and medullary region to trap the antigen which is brought by the lymph, and the lymph will leave out of the lymph nodes through the efferent lymphatic vessels. So when they leave, it will take the antibodies which are secreted by the plasma cells in the lymph nodes against the antigen that enters the lymph. Sometimes we can see visible swelling of the lymph nodes in our body, and they are nothing but the response, the active immune response which is given for the infection. And because of that, there will be an increased concentration in the lymphocytes in these lymph nodes, and so the lymph nodes will swell, and the swollen lymph nodes may be a signal for the infection. It gives the signal for the infection, and there are several groups of lymph nodes in our body. Most frequently enlarging lymph nodes are found in the neck region, chin region, in the armpit and the groin region. Now the cells of the immune system. The immune system is composed of many interdependent cells, and they help us to protect our body from the microbial infection and also prevent the growth of the tumor cells or the cancer. All human blood cells are derived from the pluripotent hematopoietic stem cells of the bone marrow. This we have already seen. And these stem cells has the capacity to produce all type of the blood cells like RBC, WBC and platelets. Out of these, the only cells that are capable of producing specific immune response are the lymphocytes. The other type of blood cells are used for producing non-specific immune response, antigen presentation and cytokine production. What is the cytokine? It is nothing but the proteins which are produced by the lymphocytes in response to an antigen in the body. Here are listed, tabulated the number of cells which are present per ml of our blood. The RBCs, they are present, they number about 4.2 million to 6.5 million per ml. Similarly, the white blood cells and the platelets are also listed. The white blood cells are classified into agranulocytes and granulocytes. Lymphocytes and monocytes are coming under the agranulocytes, and the neutrophil, basophil, eosinophils are coming under the granulocytes. Their number and percentage is also given in this table. We have to learn it. Lymphocytes. There are three types of lymphocytes. They are B lymphocytes, T lymphocytes and natural killer cells. Again, this T lymphocytes is classified into cytotoxic T cells, helper T cells and regulatory T cells. Phagocytosis is carried out in our body by the monocytes and dendritic cells. The lymphocytes have receptor proteins on their surface. When the receptors of the B cells bind with the antigen, the B cell become activated and it will divide rapidly to produce many plasma cells. It is like taking avadaram. So B cells, when it finds the antigen, it will club with it and it divides to produce more number of plasma cells. Now the cells of the immune system. Cells of the immune system are produced from the pluripotent stem cells of the bone marrow, and this will have the precursors. Here we can see the fate of the cells. The pluripotent stem cells will be forming the lymphoid progenitor and myeloid progenitor. This myeloid progenitor will give rise to the erythroid progenitor, which will be converted into erythrocytes, and the myeloid progenitor will divide to form three types of cells, that is basophil, platelet cells and eosinophil cells. Here the lymphoid progenitor will produce the B lymphocytes and the T lymphocytes, and granulocyte monocyte progenitor will produce neutrophil, monocyst and the dendritic cells. The monocyst will become the macrophage cells. So macrophage and the dendritic cells are produced from the granulocyte monocyte progenitor. Lymphocytes. About 20 to 30% of our blood cells are lymphocytes, and these cells will have a large nucleus which will fill the most of the cell, and they have only a little cytoplasm that is surrounding the nucleus. The two main types of lymphocytes are B lymphocytes and T lymphocytes. and both the lymphocytes are produced in the bone marrow, but the B lymphocytes will stay in the bone marrow until they get mature in the bone marrow itself. Some of the B cells will remain in the blood, while some will accumulate in the lymph nodes and the spleen, and they will circulate in the blood, whereas the T lymphocyte will get mature in the thymus gland. They are produced by the bone marrow, they leave the bone marrow and get matured in the thymus gland. Once the T cells mature, they will accumulate in the same area of the body as the B cells. Lymphocytes have receptor protein on their surface. When these receptors come in contact with the antigen, they bind to the antigen. The B cells will bind to the antigen, and B cells become activated after binding with the antigen, and it divides rapidly to produce many plasma cells. And these plasma cells in turn will produce the antibodies. Some B cells do not produce antibodies, but they become memory cells. And these memory cells are responsible for the secondary immune response, as we have seen before. And the T lymphocytes will not produce any antibody, only the B lymphocyte will produce the antibodies, and T lymphocyte will recognize the antigen presenting cell and destroy the antigen which are coming into our body. The two types of T lymphocytes are helper T cells and killer T cells. These helper T cells will release a chemical substance called cytokine. This cytokine will activate the B cells to perform its function. And the killer cells will move around the body and destroy the cells which are damaged and which are infected by the pathogens. Apart from these T cells and the B cells, we have the neutrophils and the monocysts. They also destroy the foreign cells by the phagocytosis. The monocysts, when they mature, they become a large cell and they are called as macrophages. And this also performs the phagocytosis on any foreign organism.